James DiCarlo is our next speaker, and I learned this morning that he was an undergraduate biomedical engineering student at Northwestern when I joined the faculty there. And it made me realize that I must have been around for a long time, because Jim's now the head of the McGovern Institute for Brain and Cognitive Sciences at MIT. You're not? The chair of the department, sorry. Yeah, it's all the same to me. Way to go, Jim. You really advanced from your undergrad BME degree. But it also shows you a great background you get with biology and math as a bioengineering undergraduate. So for me, that I'm a big fan. Jim studies the fascinating field of how we do visual recognition. And he's good. That's the theme of his talk today, is reverse engineering visual intelligence. So without any further ado, please welcome Jim to the podium.
Okay, thank you. Okay, thank you. Thank you for the introduction. I want to thank Bill Newsom and others for inviting me to this fabulous conference. I'm going to try to give you our particular take on the intersection of AI and neuroscience. Can I get the first slide up, please? Is that my slide? Oh, there we go. So I titled this Reverse Engineering Human Visual Intelligence, and I hope you'll see, this doesn't say AI or doesn't even say neuroscience, but I hope you'll see how this approach leads you naturally to an intersection of AI and neuroscience. So reverse engineering to me goes like this. So there's a goal that we have. So goals are important to us. Try to account for every ability of the mind, which I'll broadly call intelligent, complex behavior. That's intelligence is a hard thing to define, but we can measure aspects of complex behavior. You'd like to do this using only, for us, components that we know exist in the brain, things like neurons and their connections. And you want to be able to specify your descriptions in the language of engineering, which right now means models that can be implemented in machines that serve as our hypotheses for what's going on in the behavioral domain of interest. As my colleague at MIT, Josh Tenenbaum, likes to say, another way to translate this is, let's do science like engineers. And this is my reverse engineering team at MIT, and everything I'm going to say is all attributed to them and others that have gone through the lab. I'm just an ambassador of these great men and women and what they do. I want to especially highlight Dan Yeamans, who was a postdoc in the lab, and you're lucky to have here as a professor, assistant professor, and I will highlight his work, importantly, along the way. So here's reverse engineering to us as a recipe. It goes like this. So first you have to define what it is that you're trying to understand. What aspect of intelligence are you after? So operationalize some rich behavioral domain of interest, things that can be measured. Now, you want to do this in a way that makes the problem interesting from the start. So it's nice to ask, what are ways that human or animal behavior is already surpassing current engineered systems? This could be surpassing in terms of performance, which is naturally what you think about, but it could be in other things too, like power and size and other ways that biology has surpassed current engineered systems. Now, you got to keep going. This is an important key step, as you'll see, but then go and get some measurements of the internals of the system that are likely relevant to that interesting behavioral performance. And here you have to choose wisely, and this is the art of doing science, because there's many things you can measure in the brain, or behavior, spikes, anatomy, blood flow, neural perturbation, genetics. It can go on, and neuroscience is a very big area. So you have to choose wisely with respect to your goals here. And you can't stop there. Then you have to go ahead and try to build some model or set of models where you're trying to build those models under the constraints of measurements from the field that bear on this issue. So you can engineer something that can not just capture those measurements, but should be able to predict held out measurements of at least the same type that you're collecting here. So prediction being an important goal. And if we're neuroscientists, many of us care about spikes. They're an important measurement, an important information transmitter in the brain. That means that we need to build things that have neurons in them that can then predict neural phenomena like spikes. So that leads you naturally to building with things that are called artificial neural networks, that at least have elements that can be mapped to the things that you're measuring in the brain to fulfill this goal here. And if you're going to explain this complex behavior, it's also important that you're going to have to not just build toy models, but models that are built at sufficient scale to really engage on the challenges of the problem. So these are really engineering challenges that are critical to making success in this whole endeavor. You can't just stop there. The models then inform the next kind of measurements that you want to make from the system to more sharply define the models, and then you go and make new models, and the faster you can make this go, I would argue that leads towards models that will constitute our understanding over the domain that you originally specified. And of course, you can loop back and expand the domain. I'm going to return to this word understanding. It came up in Surya's talk at the end of my talk here, because I think this is quite provocative at some level. The understanding that we get out of this is the kind of understanding that is very nearly close to many applications, and I'll refer to that during the talk as well. Classically, what I'm showing you here is the interaction between what we call science, which is usually natural science to make measurements, and what I'll refer to as modeling, which here in this audience, we're really meaning things in AI, machine learning, and computer vision for us particularly. So these two fields are really, if you broaden this out, science is engaged on one of the greatest questions of humankind, which is understanding natural intelligence, whereas engineering is currently engaged in one of the greatest engineering challenges of this time, which is to engineer artificially intelligent systems. And marrying those things together is really the kind of the goal of what this is about here, and a problem I'm going to operationalize for you. But more broadly, MIT is engaged in that effort with something called the quest for intelligence. Now, so for me, I'm going to tell you about our particular approach of marrying science and engineering around the problem of core visual object perception, one aspect of human intelligence. Okay, so we started in 2002 saying we'd love to understand how all of vision works. Of course, that's an ambitious goal, and I knew that's not the goal that I'd maybe be able to accomplish in my lifetime. But we said, let's try to see if we could do a sub-goal that seemed to be very important and already interesting and challenging, which is just to be able to label the nouns in the scene like this one, to say there's a car, there's a person, there's a building, this kind of category labeling task. That was the problem that we set up, so we'll call that object or category recognition. And then we looked at biology and said, when you and I look at that image, we don't digest the whole thing at once. We have high acuity, so if we're fixating right here, we really have high acuity at the center of the visual field. And the part of the brain that's involved in recognition that I'll tell you about in a minute, especially processes the central 10 degrees. So that's what this window is meant to illustrate here, a 10-degree window. But you don't just absorb this whole scene at once by staring at this one point, but you make movements to explore the scene called saccades. These are very rapid eye movements. You don't notice that you're making them, and you don't see during them. You see here, and again here, and here, and here, and here, and you dwell at each of these points called fixations for a few hundred milliseconds. So now when you take that together, you realize that what you end up with, now operationalized by how primates digest the visual world, is something that looks more like this, 200 millisecond snapshots of the visual scene of about 10 degrees that look something like that. Now I hope that if I'll play that again, you could recognize one or more objects in each and every one of those very brief glimpses, even though you no longer see the whole scene and no longer have the scan path, you can still say there was a sign, there was a person, there was a building, there was a car. That ability, that we call that kind of operationalized as central 10 degrees, 200 milliseconds viewing duration, reporting object category, that's what I'll refer to as core object recognition. And this is my attempt to biologically define a problem of interest for you here so that we can actually make progress on it. In the lab, we actually test this not with just natural image patches, which we do, but also images like this that we can construct, where we have control over all the latent variables of the object, such as this is a car, obviously, so we know the category, but we know also its latent position, we know its position, we know its pose, and so forth, placed on a naturalistic background. And this, when you do this, it generates funny images. You know, it's sort of natural, maybe a little scary, or maybe images like this that you wouldn't see in something other than like a horror movie, but you still could recognize that that was a head or a face. And humans do this quite readily, and we did this because machines at the time were brought to their knees by even these kind of rendered situations here. And so we can test this in the lab by doing things like this, where I'll just fixate there and you saw an image and you're like, oh, I have to choose among two objects. I didn't have to tell you what you might see. You have two choices here, and I hope most of you would notice that you saw something that was more like this than like this. And here we go again, and then this was a bird, right? So we just view these as tokens to indicate your choice, and there's no pre-queuing here, and you can do this all day long, and in fact, humans, this is human performance now. Here's a bunch of humans. This is them doing an 8-way task. The details don't matter except to say performance, which is here in D prime units. Here's 98% accuracy. Very, very good for humans across this whole x-axis. What is this x-axis? This is where we introduce more uncertainty about the viewing parameters, such as the pose and so forth, and things like background variation. So this complexity here, this high uncertainty conditions where you have to generalize to new instances from the category, is what makes this problem challenging because, look, machines, at least around 2009, were very poor at doing this relative to humans, whereas they were good in these low variation conditions, right? So remember, ask ways in which this system is interesting relative to current machine systems. So that's where we were focused here in those high variation conditions. Okay, so we operationalize something, a goal that we want to try to achieve. I've been doing tests on you sort of, implicitly psychophysics as you've been watching this as humans, but I don't have very good access to the internals of your system here. So if I want to measure the internal components of how each of our brains do this, which is really the goal, I have to make a switch here to an animal model. And our lab uses this animal model, the rhesus monkey, and now I need to justify for you why this particular animal model. And a big reason we do this is we know a lot about this system, but one of the most interesting things about it is that its behavior looks very similar to our own. So here's a rhesus monkey in his home cage doing the same task you just did, triggering the onset of a new image and then choosing among two choices. And you see he does this very quickly. That means he got it wrong. But you see most of the time it's green, meaning he's getting it right. And we can get thousands of trials of many animals all day long on this. This animal knows something like 30 objects. It takes the animals about a day or so to learn each new object. I won't talk about their learning today, but their steady state performance looks like this. So this is the patterns of difficulty of discriminating 24 different objects from each other. And all I want you to see here is don't worry about the details, other than to say tanks are more confused with trucks than they are with birds. This is averaged over many images of tanks and birds and trucks. That's what's illustrated by these colors. And you can see this is true in both cases here. And I'm not telling you which is the primate, which is the non-human primate, which is the human, because we can't tell the difference between them at this level of resolution. So monkeys look like us in doing these kind of tasks. And even at higher resolution, they look like us at doing these kind of tasks. So this gives you a justification to say, okay, I've got an actual model system, not a model like a mouse model of a human, but an actual model of how we might do this in our own brain. So let's go in and get some internal component measurements from this system. There's the rhesus monkey brain, there's a human brain. So we're going to now go into this system. The reason we use this brain is not just because of its behavior, but because we know a lot about what's going on here because of a lot of decades of work of folks like Bill and others that have worked on this system for, again, many decades. So we know things like, hey, there's this place called infratemporal cortex that I'll tell you about that's up here at the end, or I'll call it IT, where lesions in this part of the brain cause deficits in that kind of tasks, things like recognition tasks. I mean, we've known that for several decades. We also know something about the anatomy here. So there's a series of visual areas called the ventral stream, area V1, V2, V4, IT. These are cortical areas. They're fed by subcortical areas, the retina and the LGN, that eventually propagate through this whole series of neurons that are illustrated here schematically. Millions of neurons in each area. And there's both feed-forward, feedback connections, and recurrent connections illustrated just schematically for you here. So whenever you see this, this is an illustration of the brain as we're schematizing it for you here, just laid out in a little more engineering format. Decades of physiology has taught us things about this, like, hey, the retina has local processing, which produces, there's a map here in the back of your retina of what's out there in the world, and each area has a new map of the visual field, and we think there's about 3 maps in IT cortex. And for the aficionados, I won't talk about that today, except to just call IT one big area for the talk today. Okay, so for decades of physiology have taught us what I've just shown you, and I'll give you another sense of what it's taught us, is that when you take an image like this one, your retina produces a nice photograph here, a photographic copy. Now, to all due respect to my retinal colleagues, there's lots more things going on inside the retina than just copying. These are important things you would have to build into nice cameras and so forth, but it's a collection of pixels that essentially captures the pixel content of the image, and it's isomorphic with the image. Now you get these neural patterns of activity that are produced in a largely but not completely feed-forward sweep through the system. In about 100 milliseconds, you get a new population pattern of activity up in IT cortex. It looks nothing like a photograph. It's a population pattern, so it's re-represented the image in some way, but in some novel, nonlinear way. These patterns turned out to be quite interesting and important. If you show a new image, you get a new pattern. If you go back to the old image, you get back to the old pattern again. So they're quite stable. But you can show that the IT cortex can follow and produce a new pattern at a rate that matches the rate that you can perform these kind of visual tasks as well. So IT can happily click along and follow this kind of input stream at the scales of your natural visual sampling that I showed you earlier. Okay, so now you have these patterns, and they're interesting, and we know they're interesting from decades of work recording from individual neurons in IT cortex. Here's an example so you can sort of see the elemental data from neuroscience of what this looks like. Each tick mark here is an individual action potential from a neural site within IT cortex. Each row is a new trial of the image. So you can see reproducibility in response to this same image presented about 10 times here. And then you see that this site prefers this image more than this. You see more spikes here and less spikes over here. You also can see the latency. Here's when the image is on and off, and then you see the latency of about a little over 100 milliseconds. Okay, so here's, again, another site. Here's another site. Different sites have different patterns of selectivity, and I'll return to that in a minute. Okay, but for fixed ideas, we need to quantify these measures. There's lots you can do here for counting up spikes, lots of interesting questions. For us, we do the most simple thing to start, and I'll back off this towards the end of the talk. We'll just average the spikes over a time window here of about 100 milliseconds and average across trials. So now for each image, we get one number. So that spike, that neuron gives you four images. I get 4 numbers, and this is just giving you a sense of that with those numbers shown here. But remember, we're not just going to collect one neural site. We're going to try to sample as many as we can. We can't sample millions of neurons, but we can typically sample hundreds, and we can pool over to get thousands or so. And we do this with these chronically implanted arrays that get implanted during a surgery that allow us to stably record from neural sites over many weeks to months. These are commonly used often in the motor system. For instance, Krishna Shenoy's lab here at Stanford. Here we found a way to get them into the ventral stream of a monkey, and we typically put 3 in at once. And then we can connect the animal every day and record from these and get a lot of data this way. And this allowed us to dramatically increase the amount of data we could get per unit time relative to standard single electrode methods, which had been used even just 10 years ago. So when you get this, what you get now is you have a neural response vector that looks like this. So now remember, these are the numbers I just showed you. So each of these is just the spiking rate. Now, these are not one neuron, but these are different neurons in response to one image. So here you see a bunch of neurons, and again, we can get to the kind of these orders of magnitudes of neurons. The color intensity just indicates how high the average response is to this particular image. I'll show here's not just one image, but here's 8 images. And we don't just collect 8 images, but as we said, we've increased our data rates so we can get thousands of images. And we use a lot of our data capacity to collect many repetitions so we can get high SNR on the response to each of these images. And that's hard to see on this here. But this gives you a sense of what the volume of data that looks like when we collect data out of say, one animal's hemisphere of IT. Okay, we knew about more than a decade ago that if you take these population spike patterns in IT and you count spikes as I just showed you, you produce a vector here in yellow, exactly analogous to the green vector I just showed you. I'll call it a feature vector. You can think of that vector as being a point in a population space that's illustrated here, where this is the activity of 3 IT neurons, just to illustrate fixed ideas. Here's the image, there's the vector. You can have a high dimensional neural space. And now imagine all the points that would arise from this neural population for a bunch of images, some of which are faces, some of which are schematically shown as non-faces. And the job of something to do a categorization task, to say face and non-face, is to separate these points. And we do that with linear classifiers, which are planes in 3 dimensions, hyperplanes in higher dimensions. And the basic idea is it partitions the space in such a way that if you see a future image that causes the vector to live on this side of this space, the system will naturally generalize, even though that's not an image of a face you've ever actually seen before, to still report, aha, I think it was more likely a face than not a face. That's what these kind of systems do. And if your representation is powerful, you can just use linear tools to enable this. So this is called a linear classifier. It's exactly analogous to the perceptron that Bill introduced at the beginning, which you can think of as a weighted synaptic sum on some downstream neurons with a threshold. And these are mathematically, these are exactly the same idea. To summarize more than a decade of work, what we know is that if you take linear decodes out of IT with relatively low numbers of training examples, that you accurately recover these kind of behavioral patterns that you see in both humans and non-human primates that I showed you earlier. So you have this sort of simple linear linking mechanism that goes from here to here. And for the aficionados, of course, the specific parameters in here are really interesting if you're going to try to do things like inject object percepts by perturbing IT neurons that would cause predicted changes in behavior. But I'm not going to have time to tell you about those experiments or those parameters today. Instead, this is all to just set up for you that there is a powerful set of features, a general purpose basis for vision, or at least core recognition, that lives here in IT cortex. So it's the kind of penultimate product, penultimate before behavior of the brain's processing stream to enable tasks like recognition. And that's all I want you to take from that long introduction. And this explains this gap here. So why are humans so good? It's because they have this powerful set of features that with linear decodes naturally brings you up to high performance that's highly tolerant to uncertainty here. So the magic is in the feature set, and it's relatively simple to get to behavior. Okay, so you might say, well, okay, that's nice, but this is not fully a satisfying answer if you're doing reverse engineering like I said I wanted to do. Why is it not satisfying? Because I sort of said there's a magic set of features up here in IT, but I didn't give you an end-to-end specification that goes from the image up to these features so then I could then get to the behavior that I defined at the beginning. I just sort of told you you can go from here to there. How are these features computed? That was a holy grail question for us for a long time. And similarly, what are the intermediate features along the way that I've illustrated here? Now, what you may know, and Bill introduced this, is we really had a breakthrough in this around 2013, and I'll show you that work today, but this is still a continuing question of how we do this. But we had a breakthrough in our understanding to go from images to these IT responses, and set up by an understanding of how IT relates to behavior. Okay, so even just 5 years ago, we didn't know what IT neurons liked. We didn't know what they were really doing. So they were enigmatic. You could think of them like Egyptian hieroglyphs. We did before we had the Rosetta Stone. So the idea was you have a bunch of IT neural spiking responses, and I showed you some of these examples earlier, but now I'm showing you for these complex images and many more images. So here's 1,600 tested images, and again, high SNR, and these are mean spike rates of one site in IT. And here's, again, just 4 example images. These happen to be of chairs. And you see these are different chairs under different renderings and so forth, as I mentioned earlier. And you can kind of squint at this plot. And if you squint up your eyes, you could say this is a chair neuron because it responds more here than here. But that's not fair to the poor IT neuron, I would submit. Why is it not fair? Because, well, it doesn't really like that image of the chair, and it likes this images of the planes and something about a boat here. And you might start thinking it's something about lines or edges or something like that. And this has gone on for decades where people try to decipher what was going on. Here's a classic neuron out of IT, so-called face neuron. Face neurons were first studied by Charlie Gross and later by folks like Marge Livingston and Doris Sau and Vinrik Frywald. And face neurons, this is a classic face neuron. Look, it responds more to images of faces than to other things here in the set. And these neurons are not, again, it's not fair to the poor IT neuron to call it a face neuron because it likes that face but not that one. And for some reason, this image of a plane it really likes in this table too. And so it's doing something more complicated than that. How are we going to understand that complexity? Well, this is where the engineering helps us out. So I've just showed you measurements. I kind of showed you why IT is important and set up we need to understand those. But to get there, we have to build some more models. The models that I'm going to show you, they were engineered to try to just digest the central 8 degrees here. So they're not doing a full behavior, full reinforcement learning like you heard in the previous talks. It's just trying to say, how can I process this image to a useful set of features? So just this part here. And so the models that I'm showing you, they didn't pop out of nowhere. They were they were driven by a lot of data from, again, decades of neuroscience, some of which is illustrated here. For instance, spatially local processing that's repeated over the whole space of the input, that's essentially implemented as a convolution to approximate those operations, which are textbook in neuroscience. Threshold nonlinearities, nonlinear pooling, normalization, the fact that there's a stack here that I showed you anatomically that exists, a so-called deep stack. These are things that are known from neuroscience for many decades that were put into models. Originally in 1980, a model by Fukushima called the neocognitron was the first attempt to try to model the ventral stream and how it does recognition. This was extended with a model by Tommy Poggio. Tommy Poggio's group called the HMAX class of models for nearly a decade. Later, my group with Dave Cox and Nicholas Pinto as grad students started searching through this space of models to find high-performing models within the family of models. And then we led us up to this model called HMO, which was produced, again, by Dan Yeamans when he was a postdoc and Hung, a graduate student in the lab. You might not even know HMO or hear that as a model in the field, and it's not important that you do. What's important is you understand the process of how we got to HMO and what it taught us. So I'll tell you about HMO next as a neural network model, but I just want you to see it was part of a long string of models that had already been being developed. Again, these models, like HMO, they're neural networks. So within each of these planes are thousands of simulated neurons that are basically perceptron-like units. We macro constrain the architecture, meaning we have 4 different areas that we thought would be roughly analogous to the ventral stream. We have multiple filter types in each area, which is illustrated by these kind of planes here. We put things in the models, these neural network models, where their filters are linear operators followed by some output nonlinearity with some normalization. It was already mentioned. This is inspired by ideas in the literature from neuroscience, for instance, Carandini and Heeger, Movshin and others. And we put those in as loose constraints on the model architecture. So we put a lot of things that people had thought about in the field, but we didn't try to make it perfectly biomimetic. And importantly here, we didn't fit the model, right? So we didn't take any of our neural data and try to optimize the parameters of the model to explain the data we collected. What we did instead was say, let's get this model to do a task, do the intelligent task that I said at the beginning, which is core recognition, and just try to optimize the parameters to solve invariant core recognition tasks, which again was informed by fields like cognitive science. How do we do that? Well, Dan used a bunch of applied math and computer science tricks to tune the parameters. For the aficionados, he wasn't actually using gradient descent here. We were searching through architectural parameters to optimize to do this mostly. And that's kind of where the name HMO actually comes from if you want to go read that paper. Today, people usually do this by stochastic gradient descent. To me, it doesn't much matter. I think both of these things are not completely biological. I don't pretend them to be biological. All I'm using them for is a way to get the network into an adult state that is doing the task that I think the ventral stream might do, and then I can ask about what it looks like inside relative to the adult brain that I'm measuring. So this part is not biological. These two are constrained by biology, although this is, of course, an interesting area of work that people are actively debating right now. The thing I want each of you to realize is that every time we find a new set of parameters, every time we run an optimization or stop an optimization and set the parameters of the model, we've created an entire new visual system. It's a hypothesis of how the visual system, the ventral stream, might work. So I like to think of this as us doing evolution of some form in the computer, not meant to recapitulate normal mammalian evolution, but just a form of evolution or selection within a computer that's trying to solve this task up here. But the question is, what does it evolve to? When I evolve that kind of machine, where do I end up? So here's what we'd hoped. We'd hoped that we would end up with something that looked like the brain, the ventral stream. But this seemed like kind of a long shot, although we knew this model was in the right space. But it turned out that this works remarkably well. First, before I show you how well it works, how much it looks like the brain when we evolve it this way, I tell you that we kind of have to first map this to the brain. Remember I said earlier that the neural activities need to be mapped somehow. So there's neurons in here, simulated neurons. There's neurons in here that we've measured. They don't have labels on them, but I can take data, response data from each neuron, use some of that data to find a corresponding neuron in here that maps between them, and then I can make predictions from this model as to how this observed real neuron will respond to new images. So there's a bit of neural data that's used to do what we call mapping. And for the aficionados, you'll know that we didn't actually do this neuron by neuron, although that works actually reasonably well. We did it as a weighted sum of neurons within here to try to fit individual neurons here. And we did this both in IT, the area I already showed you, and its input area V4. And again, we ask, how well can this mapping now predict the responses to held out images? And as Bill already sort of alluded to at the beginning here, it actually works remarkably well. So here's an IT site from the data I showed you earlier. Here's the same kind of chair neuron that isn't really a chair neuron I showed you earlier. Yet the prediction from the ANN model, and this is from its, quote, IT level, actually follows all this detail remarkably well. And these are all images that it never saw before. It's predicting all of this. And we can quantify all this. It's not perfect. Here's the face neuron, the sort of a face neuron, but not really a face neuron, and it follows all that detail too as well. So we can quantify both these things, and we're explaining about half of the explainable response variance here, and that's what's quantified here, relative to 20% in old models. So this is, when I said we had a breakthrough, this is what a breakthrough looked like to us, is that suddenly we were able to explain things with a neural network that we weren't able to explain before. Here's V4, the input to IT. Here's a recording from V4. And this one you wouldn't even squint at and call it a chair neuron or a face neuron or anything. It just looks complicated in its response pattern. And remarkably, though, that complicated response pattern is predicted not by the highest level of the HMO model, not by the lowest level, but by an intermediate level here. And that was also interesting, that the even more hidden units match the even more hidden units within the ventral stream. And that, again, is about 50% of the explainable variance. So big picture of what I just told you is that we kind of have this model, it's this model called HMO that was explaining about half of the explainable response variance in IT with its top layer or its sort of last hidden layer, and in V4 with one of its intermediate layers, as you see here. And compared to a bunch of control models, these were, again, big advances for us. I want to highlight that this approach worked well for us in neurons, and then people in the next few years use these same kind of approaches to do this in human fMRI studies. And I especially want to point you to the work of Nikos Krigas-Gorte on showing these same kind of approaches explain what you're seeing at the level of fMRI in the human ventral visual stream. But the summary for you guys, you should say, look, we've got about half of the explainable response variance explained at this point. So if you missed everything I just said, here's a summary of what I've told you. We evolve artificial ventral visual streams that are deep neural networks. We found that we got about a 50% match by the measures we use here. Similar match here, although it's a little bit higher at this point. Other labs, Andreas Tolias and Matthias Bethke and others have shown that this actually works pretty well in matching V1 and even better than some standard models of V1. And we also can show that there's actually a pretty good match to the behavioral level here when you just measure the outputs of the system. But I don't have time to tell you, but there's clear problems up here. It's not like these things are perfectly matching behaviors, but I don't have time to tell you about that today. So that was a big picture summary of what we had gotten to, and that was, around 2013. Here's one of the key take-home messages that we took from this work. And this is the connection between neuroscience and artificial intelligence. So each dot here is a full ventral visual stream. It's a neural network with random parameters. Each black dot is a model from the field. And the way we looked at this, and this is a plot in one of Dan's papers, is it shows performance of a deep neural network model on the invariant task. And this is the ability of the last hidden layer of the model to match primate IT in the manner I just described to you a moment ago. And you see this correlation, and so that argues that if you can optimize to do better on these kind of AI goals, that it brings you up here, and if the correlation holds, then you should explain better. And indeed, that's how we think about HMO, which we first produced in the lab around 2012, explaining about half of the explainable response variance. So once you look at a plot like this for a while, it kind of gives you an idea. Well, look, here's neuroscience goal, explain these neurons. Here's an AI goal, do good computer vision, machine learning. And you say, well, why don't we just keep doing this? We can neuroscience, sit back, wait for AI, computer vision to pump out a new model that's even higher performing, and then it should be even better at explaining the brain. And, this is what this seemed to imply to us. And I said, that can't hold forever, but let's see how far it holds. And so now you can kind of step back and look at the larger fields and say, well, look, here's brain and cognitive sciences. I described this sort of trend of models already briefly to you, trying to just figure out how the ventral stream works. But then computer vision is, of course, busy trying to do things like AI, computer vision. You'll hear some of this from Fei-Fei, and there's lots of techniques that were being used. And then there was a thread that was more of a neural networks thread that kind of wandered for a while, and Jan LeCun and Jeff Hinton and others, they weren't performing as well as other systems. And then through a combination of factors that I don't have time to explain, suddenly they were performing very well. And indeed, around 2012, kind of so-called deep learning started to take over computer vision and a lot of fields, as you know. And so then we were able to ask, well, okay, what about these models that were coming out of computer vision and these later computer vision models? They're high performing on the same kind of tasks we we were working on, maybe they're even better models of the brain than the model that we had been building. So here's now our plot again. I've changed the format on you again. This is the neuroscience fit to IT. Here's the AI goal, trying to get, this is ImageNet validation of performance. This ImageNet is an important test that Fei-Fei developed to help drive computer vision. And so you see that there's this correlation here, but we're not yet up to the max possible, again, about 50% of the way there. This model out of AlexNet from Jeff Hinton's lab was actually better at explaining IT than the model I just told you about, HMO, a minute ago. So again, it wasn't even looking at any of the neural data. We checked it against the neural data, and it was better than the model we had built. So the way you can summarize this is that for a bit, AI is doing the work of neuroscience as it engineers these models within some architectural space. It was actually producing neural networks that were better matched to the brain. But this can't hold forever if you think about it for a while. And in fact, it doesn't seem to be holding anymore. So here's more recent models. They're performing better. So they're doing the job of AI or computer vision. Yet you see, at least in this prediction of IT, which is just one time window within IT, this is sort of flattening out. So we can't just keep waiting around for AI to kind of do our work for us as a neuroscientist. Okay, so here's a summary, if you missed everything I told you, what I told you so far, is that after a while, several deep ANNs artificial neural networks behave both internally, and I showed you some of that, and externally, the behavior, I only mentioned that, far more like primate brains than any previous artificial systems that we had before. So you should not lose sight of the fact that this was a remarkable advance, but I'm showing you this half full, half empty, because of course, there's still problems. But I'm going to talk about those, but I want to point out that these advances were made possible by ideas from brain science, anatomy, cognitive science, development plasticity. I tried to introduce some of those along the way, but with a strong dose at the end of taking these same ideas and injecting engineering in more of an AI machine learning framework to actually do the optimization. And so it's this marriage between them that's led to this big sort of filling up of the glass this far so far. But no deep ANN has passed our tests. 50% explained, not fully explained. I'm at 5. Okay, so I'm going to go quickly now. So I'm really only at 5? Okay, so summary. Our field has achieved a good understanding of the initial ventral visual stream processing, not its learning, not its spatial layout, and one of its intelligent abilities. What can we do with this understanding? Well, I'll just tell you briefly, one of the things that we can do is we can start to control neurons, or try to control them. So if you build a model, you map it to a neuron, you can then go back through the model, synthesize an image that attempts to put the neuron in a state that you desire. You can do this for individual neurons or for populations of neurons, and this is something, active work in the lab right now, still unpublished by these 2 postdocs. And one of the goals, you can try to do things like, here's a V4 neuron, let's see if I can drive it well beyond its previously measured firing rate. Here's the predicted firing rate from a model that had been mapped to it. Look how good this correlation is. That's just recapitulating what I told you earlier. But we can search for images that are synthesized that should drive it even better than its best naturalistic image shown here. Here's the model trying to drive this neuron up to a high firing rate by generating these crazy things that look like nothing, but they're kind of cool to look at. And what's remarkable is that these synthetic images predicted to drive the neuron high actually do drive the neuron high. So this is evidence that the model is actually good in some ways, and it sort of starts to say we can start to use external images to control the neurons. What's really more exciting to me is not just a single neuron, but you can start to think about a group of neurons as a control target. So imagine you had a whole bunch of recorded V4 neurons, and your goal was to turn one of them on and have all the others off. Could you construct images that could do that? If you search through natural images that we had tested, this was the best we could find for this particular set of V4 data. And you see it does this pretty well, but there's all this off-target activity. And that's not surprising because the receptive fields are strongly overlapped on the visual field. So energy coming in here is kind of, is having all these off-target effects. But the model produced some image that, I don't know, looks like nothing, but somehow is trying to control this neuron. And remarkably, it's sort of able to do that much better than the images when you search through here. Now, these are still early days. This is just one example, but it gives you a flavor of what you might be able to do with these things. So what can we do with this so-called understanding? I think you can do a lot of things already, right? Non-invasive neural control, non-invasive perceptual control, invasive perceptual control if you inject things, front-end models for memory attention, generate hypotheses for disruption. You can do a lot with this so-called understanding. The only thing I know you can't do is you can't always make a bunch of scientists happy that this constitutes understanding, right? So, and I think we'll talk about that at the end. So I think I have about 2 minutes left. Is that about right? 1 minute? All right, so I'm going to skip through the fact that, wow, I thought I had timed this out. So we want a model that explains many things. So we built a brain score that is capturing many of those things. And this just recapitulates what I showed you earlier, that the brain score, AI improves, the brain score is generally going up, but more recently it's sort of flattened out. And we need something else besides performance to guide us now. If you like brain score, go to this site. I'm going to skip through this. This deals with recurrence in models that I tried to sneak in, but I obviously have no time for. We built recurrent models that are now achieving higher brain scores than any of the deep neural networks that we had been testing so far, or on par with them, yet have much simpler feed-forward simplicity properties. So we're thinking if you want to proceed on this, we need to start putting in other constraints besides performance, things I mentioned at the beginning, wiring costs, size, and power. And if you put those in, you'll converge the things to be more like the brain, and this Cornet is an example of that in an archive paper from our lab. But I just want to end with this provocative part because Bill said put something provocative in at the end. So how can we discover more accurate models, get more measurements, and use them as constraints? So here's the provocative part. So the classic way of doing science or neuroscience goes like this. You pick a task, maybe, you operationalize it, you make a bunch of measurements, and then some smart neuroscientist that's trying to be Einstein says, here's understanding, and you write that understanding into the abstract of your paper, and you communicate it to other humans, and if you're lucky, you get someone to help you model that phenomenon, and this is sort of what passes as understanding. That's not going to work for complex systems. That's the main thing I'm submitting here. The reverse engineering recipe changes this old neuroscience model in 3 ways. First, it uses AI or artificial neural networks to actually optimize for a task of interest. That's what I took you through in my talk. And that puts the models almost in the leading edge of the hypothesis generator, as I showed you there. The second thing it does is it moves the word understanding from the abstracts of papers of neuroscientists to the model itself as the encapsulation of our understanding of a complex system. And this is the part that I think is worthy of discussion and debate. And then the last thing it does is it puts you in a mode where you say, I need to turn that crank between the measurements and the model as fast as I possibly can. And our brain score is an attempt to try to take our measurements and apply them to generating new models as cost functions. So not just to tell you recurrence is important, but to give you a bunch of neural constraints on which recurrent models are actually the better models according to our measurements. And right now, that crank turns very slow over months or years. Even this is how we go inside my lab. And so now the third thing you want to do is try to get the humans out of the loop, so to speak. Put the humans on the outside managing the searches of the models and the data as it's being generated as costs onto the models without trying to interpret the data as a human in the middle. And that third thing will allow this crank to spin much faster. So this is really trying to automate the process of getting towards these models from the brain data. So those are the 3 changes that you want to make. And the last thing I'll say is if you like this, plug in your domain over there. It doesn't have to be core recognition. Port in whatever you think is interesting, and you can follow the same path. And I think you'll be pleasantly surprised by where you might end up. So time's up. Thank you.